Hello, hi everyone. My name is Vishali. Today I'm gonna discuss about what immunity is. Every one of you may be aware of this term immunity because of this ongoing pandemic. So I thought why not to make a video on this what exactly immunity is.
Immunology is the science that deals with the immune system. The word immunity is derived from the Latin term immunis, which means exempt. So, immunity is the ability to fight against any bacterial infection, that's viral or bacterial or any other. So, this is a kind of a defense mechanism of our body. See, every organism have their own defense mechanism to fight against any organism that's gonna harm them. So this is the defense mechanism of our body which fight against any foreign particle that's gonna invade in our body and may it's like harmful to us. So immune system constitute of cells and tissues and organs. So further I'll brief you about cells and the organs. There are two types of immunity. First is innate and second is adaptive. So let's begin with
Innate
the innate
immunity is present since birth. It is non-specific and no memory. Non-specific means it does not have diversity for specific antigen. That means it encounters every pathogen that invade in our body. And no memory means it does not have memory cells that remain in our body that help to recognize the infection which was already there or is having second time. Barriers are the first line of defense like skin, epithelial layer, hydrolytic enzymes like lysozymes. So skin is like the biggest barrier, that the first line of defense. See, skin have a sebaceous gland that protect from organisms. So innate immunity that becomes active only entering the foreign particle. So the molecules of innate are complement proteins, cytokines, pattern recognition molecules, acute phase proteins, cationic peptide and enzymes. So they are phagocytic in nature. Not all molecules involved in the innate are phagocytic, but most of them, like phagocytic are the example of the phagocytic is macrophages that engulf the foreign particle.
Complement means to amplify the action of immunity.
They are glycoproteins. They are made up of 30 serum proteins, or they are a cascade of proteins that create pore on the membrane of pathogen that leads to the destruction of pathogen. They forms a bridge between innate immunity and the adaptive immunity. That means they crosstalk with the innate and the adaptive immunity. Whenever the infection rises, that innate immunity doesn't control the infection, then they crosstalk with the adaptive and they work hand in hand. Next is cytokines. The low molecular weight proteins that released by a particular cell population, that's only one cell population, act as an intercellular mediator, and it includes monokines, lymphokines and interleukins. Cytokines, they are interleukins and released by macrophages to produce more acute phase proteins. What are acute phase proteins? We'll discuss on the next slide. Interferons, they are the class of cytokines that made by cells in response to virus infection. And chemokines are positively charged proteins. They guide the migration of white blood cells to a target protein. Recognition molecule actually recognizes the pattern on the pathogen with the help of recognition receptor, that is called as pattern recognition receptor or PRR. There are some classes of the pattern recognition receptor like toll-like receptors, nod-like receptor, C-type lectin receptor, but the most important is toll-like receptor. Toll-like receptor is a transmembrane protein which acts as a signaling receptor. Its N-terminal is leucine rich that helps
to recognize pathogen associated molecule pattern, and C-terminal, that is a domain that leads to the further downstream signaling. TLR is 10 members in human and 12 in mouse. So TLR, that is toll-like receptors, they are further classified into surface TLR and intracellular TLR. So human surfaces TLR are 1, 2, 4, 5, 6 and 10, and the rest are intracellular. Next is acute phase proteins. They are a group of plasma proteins. So they stimulate by the microbial infection. So they include C-reactive protein, serum amyloid protein, mannose binding, which is a second type of immunity. It is a delayed immunity. It's not an instant immunity like innate. It is antigenic specific. Innate was not specific. It is immunological memory. Innate has no memory. It has a diversity, and they can very well differentiate between self cell and non-self cells. So the cells involved in adaptive immunity is lymphocytes. It's T lymphocytes and B lymphocytes. So T lymphocytes, they further divided into T helper cells and T cytotoxic cells. T helper cells have a receptor on the surface, CD4, and cytotoxic cells have a CD8 receptor on the cell surface. T helper cells actually helps other cells to grow, like B cells, and cytotoxic cells, they secrete the chemicals to fight against the infection. B cells make specific antibody against specific antigen. So innate and the adaptive crosstalk with each other. Adaptive immunity is of two types: active adaptive immunity and passive. Difference between active adaptive immunity and passive adaptive immunity. Active adaptive immunity activate naturally by exposure to pathogen or by vaccination. In vaccination, we give attenuated antigens that leads to the activation of immune system. And in passive, there is a transfer of immune cells to non-immune person. In active adaptive immunity, person develop his own immune response, and in passive, antibodies have been transferred outside the host body. Time taken by the active is weak and passive is immediately. Why time is taken by active is more as compared to passive? Because in active adaptive immunity, there is the activation of the immune system that leads to the activation of the cells, then organs, and this is a long process. But in passive, we give the antibodies from an immune or a recovered person to a non-immune person. Immune system activate in case of active and not activated or no need to be activated in case of passive. Why activated? Because as we know, natural phenomena leads to the activation of immune system to fight against any foreign infection. Memory in active is yes and in passive is no. Memory in active is yes because immune system worked against a particular virus or bacterial infection, and they have an idea, they have a memory. Yes, we have fought against this infection earlier also, and in passive there is no such case. In coronavirus, we all know that there was plasma therapy going on. Actually, plasma is rich in antibodies against any infection. So in case of coronavirus, plasma was active in antibodies against the coronavirus. So what they were doing, they were transferring the plasma from the recovered one to the infected one. That means immune to non-immune. That is a case of a passive immunity. Israel is also working on the artificial passive adaptive immunity by producing the antibodies in vitro. Antibodies produced outside the host and then introduced within the host. So they are working in vitro, that is called as monoclonal antibody, that they are working on a particular antigen that leads to make particular antibody. Between the innate and the adaptive immunity. Innate immunity is present since birth, and adaptive is after birth, like exposure to infection. Specificity, they are non-specific, and adaptive, they are specific. Like they have a diversity to the different antigens. Time in innate is rapid and adaptive is slow. Diversity, that's limited in innate, and adaptive, they have a high diversity. Memory against foreign particles, that is absent in innate and that is very much present in adaptive. Cells that involved in innate are phagocytes, natural killer cells, macrophages, and adaptive, that's it, lymphocytes, T cell, B cells, antigen presenting cells. Antigen presenting cells we'll discuss on next some other time. And distribution, they are vertebrates and invertebrates in innate, and adaptive, that is vertebrates and that too jawed vertebrates. Thank you.